everyone, and welcome back to Willow's Notes. In today's video, we will talk about the regulation of the cell cycle. Researchers wanted to know what controls the cell cycle, meaning how does the cell know that it's time to progress into S or into G2 or even to start mitosis. They wondered if there are molecules within the cytoplasm that control this progression. To investigate, they conducted a lot of experiments, and today we're going to go over 2 of these experiments. In one experiment, they fused a cell in the S phase. Remember S phase? It's the phase in interphase where DNA replication takes place. So they fused it with a cell in the G1 phase, in the gap 1 phase where the cell is still growing, DNA replication hasn't started yet. The nucleus of the cell, which was still in the G1 phase, immediately entered the S phase and it replicated its DNA. In another experiment, they fused a cell in the M phase with a cell in the G1 phase. Now, a cell in the G1 phase has to go through S and G2 in order to start the M phase. However, when the cells were fused together, the G1 cell immediately switched and started mitosis without even replicating its DNA. So, what do these experiments prove? They prove that there must be molecules within the cytoplasm that control the cell cycle. When the cells were fused, those molecules were present in the S or in the M, but not in the G1. And after fusion, the molecules diffused into the G1 cell and signaled the nucleus to begin the S phase or to begin the M phase respectively. So there must be a cell cycle control system that directs all the events within the cell. The cell cycle is controlled at checkpoints. There are 3 important checkpoints. One at G1, another one in G2 towards the end of G2, and one in M. In these checkpoints, the cell checks if everything is okay to progress to the next phase. And once everything is okay, they give the green light, then the cell progresses to the next phase. These checkpoints are controlled by proteins, by regulatory molecules, okay? Now, we're going to start with the G2 checkpoint, and specifically with the kinases and the cyclins. Kinases are enzymes that phosphorylate, meaning they add a phosphate group on proteins to either activate or inactivate them. The kinases themselves are inactive, and they need to be activated to do their function. Here comes the role of the cyclin. Cyclins are proteins, the levels of which fluctuate within the cell and hence the name. Now, when a cyclin binds to the kinase, it activates it. And together, the cyclin and the kinase form what we call Cdks, or cyclin-dependent kinases. The very first CDK to be discovered was the MPF. MPF stands for maturation promoting factor. To help you remember it better, think of it as M phase promoting factor. Because if you look at the figure, you can see that MPF activity peaks at the M phase. Why? Because it triggers the cell to pass the G2 checkpoint and proceed into the M phase. As you can see here in the graph, the cyclin concentration drops in the G1 and in the S phase. But at the end of the S phase and all through G2, it starts to increase. The levels of the kinases don't increase and decrease. They're always there. But because the cyclin is increasing, the accumulation of the cyclin will create the CDK complex, and now the CDK or the MPF activity is at its highest towards the end of G2. And since MPF is now the active form of the kinase, it phosphorylates and activates the enzymes needed for mitosis. In anaphase, the cyclin of the MPF is degraded, and the kinase is released, and it is recycled. In the absence of the MPF complex, the cell terminates the M phase, and it goes into the G1 phase. The G1 checkpoint is also called the restriction point, because if it receives the go-ahead signal at G1, the cell will proceed in its phases and divide. But if it doesn't, then the cell goes into a non-dividing state called G0 phase. Some cells, once they switch to the G0 state, they stay at G0. For example, our muscle cells and nerve cells, they switch into G0 in the mature human, meaning they stop dividing. That's why our muscles and nerves get old as we get old. Other cells can come out of G0 if needed. For example, liver cells, in case of injury, can come back from G0 with the help of external signals, of course, like growth factors, and they start to divide. What about the M checkpoint? The M checkpoint basically checks if all the chromosomes are properly attached to the spindle fibers at metaphase. Here, other key players are involved and not the cyclin-dependent kinases. If all is checked, then these regulatory proteins activate the enzyme responsible for separating the sister chromatids during anaphase. This is it for today's video. I hope you found it easy and useful. Don't forget to give me a like and leave a comment, and I'll see you in the next one. Bye!